The objective of this practical is to demonstrate the differential count of leukocytes in human blood. Now we will understand the principle of this practical. Blood is a highly specialized connective tissue which is composed of cells and plasma. These cells consist of red blood cells, white blood cells, and platelets. White blood cells are the first line of defense in human body. They are irregular in shape and basically divided into two groups. First is granulocytes, which contain granules in their cytoplasm, and second group is agranulocytes, which do not contain granules in their cytoplasm. Now we will talk about granulocytes. They are further divided into three types on the basis of staining power of granules and size and shape of their nucleus. First type is neutrophils. The color affinity of neutrophils is neutral, that is, combination of both red and blue. Their granules are very small in diameter, and these granules take both acidic and basic dyes. Neutrophils are the most visible cells. They separately give ground glass appearance. These neutrophils are polymorphonuclear, that is, more than two lobes, mostly 3 or 4, are present in their nucleus, and the connection between these lobes is very thin. Second type is eosinophils. They take acidic or red dye. Their granules are bright red colored and densely present in the cytoplasm. Their nucleus is of red color, bilobed, and connection between these lobes is slightly thicker. Their nucleus is spectacle shaped. Third type of granulocytes is basophils. They take basic or blue dye. Their granules are deep blue colored and densely present in cytoplasm. Their nucleus is blue colored and bilobed. It is overlaid with coarse granules. The connection between lobes is very thick, which is not clearly seen. Now we will talk about agranulocytes. They are further divided into two types which differ in size and shape. First type is lymphocytes. According to their diameter, lymphocytes are divided into two types, small and large lymphocytes. First we'll talk about small lymphocytes. The nucleus of small lymphocytes is so big that cytoplasm can only be seen in one corner. Stains are deep blue, giving a pink spot appearance. Their cytoplasm is crescent in shape. Second type of lymphocytes is large lymphocytes. Their nucleus is dark blue. It is large and round, almost covering the whole of the cell. The cytoplasm of large lymphocytes is light blue, which is seen as a ring in the periphery. The amount of cytoplasm in large lymphocytes is larger as compared to small lymphocytes. Second type of agranulocytes is monocytes. They are the largest in diameter. Their nucleus is dark blue in color, kidney shaped. towards the periphery of the cell. Their cytoplasm is light blue in color. What are the requirements for this practical? In order to perform this practical, you will need a microscope, blood lancet, spirit or alcohol swabs, dropper, a drop bottle and a wash bottle of distilled water, fluff-free blotting paper, sufficient number of dry, grease-free standard glass slides, and Leishman's stain. Now we will talk about Leishman's stain. It contains a compound dye, eosinate of methylene blue, dissolved in acetone-free methyl alcohol. Eosine is an acidic dye which stains the cytoplasm pinkish, while methylene blue is a basic dye which stains the cytoplasm blue. Methylene blue stains the nuclear chromatin blue-violet. The function of alcohol is to fix the blood. What is the procedure of this practical? The procedure of this practical consists of 3 steps. First step is preparation of smear of blood. Take 2 slides A and B. Both of these slides should be totally cleaned from dust particles. Then prick your finger and discard the first drop of blood. Then allow a big drop of blood to form and touch it on the corner of slide A. Then hold the slide B in your hand between thumb and index finger at an angle of 45 degree with slide A. Now place the slide B on slide A and move it towards the drop of blood till it touches the surface of the drop of blood. Now move the slide B in opposite direction very quickly and smoothly. A very thin layer of blood will be obtained on the surface of the slide A. This single layer of cells is called smear. Now keep the slides in the same position for about 15 to 20 seconds. Let them dry, otherwise cell membrane will rupture during the staining process. Second step is the staining of the slide. Pour Leishman's stain on the slide. After 2 minutes, add twice the quantity of distilled water to the stain. Mix the stain and water. Wait for 10 minutes. Then wash the slide in fast flowing tap water for 1 second. Allow the slide to dry. Then see it under microscope and identify the cells. The third step is the counting process. Add 1 drop of cedarwood oil and observe the slide under oil immersion lens. Then make 100 squares on a paper. Then differentiate and count white blood cells and fill up the squares. Move the slide either in the horizontal or vertical direction till 100 white blood cells have been counted. The percentage distribution of each type of leukocytes is then determined. Knowing total leukocyte count, the absolute number of types can be calculated. Now look at these blood films. This is how a blood smear looks. This is the tail end, this is the head, and this is the body. Now look at this side. This is the examination area towards the tail end where you differentiate and count different types of white blood cells. What are the observations? All the cells of the blood are selectively stained and spread out in a single layer. They are clearly seen under oil immersion. In the blood tissue and in tissue cultures, the white blood cells show active amoeboid movements. Three types of cells are seen. First is red blood cells. The red blood cells are stained orange-pink. They are of uniform size and they are evenly spread out. The white blood cells are larger than red blood cells. They are unevenly located. Their nuclei are stained deep blue-violet and it may appear as a single mass like in lymphocytes and monocytes, or it may appear as lobed like in granulocytes, while the cytoplasm is stained a neutral color, that is brick red or blue. The platelets are membrane-bound round or oval bodies. They lie here and there in aggregates of 2 to 12. They stain pink-purple. Being fragments, these platelets have no nuclei. Next is the identification of leukocytes. A leukocyte can be identified on the basis of its size, its nucleus, and the cytoplasm. First, note the size. The size of a white blood cell is assessed by comparing it to the size of the red blood cells. Then note the nucleus. Note whether the nucleus appears as a single mass or lobed. If the nucleus appears as a single mass, then note its shape, that is, whether it is round, oval, or kidney shaped. Also note that whether the nucleus partially or almost completely fills the cell. Then note the cytoplasm. The cytoplasm may not show visible granules like in lymphocytes and monocytes, or these granules may be very fine and of a neutral color, that is light violet, taking up both acidic and basic stains like in neutrophils, or these granules may be large and cause staining brick red like in eosinophils or deep blue like in basophils. Also note the amount of cytoplasm in relation to the size of the nucleus. This is how different types of white blood cells will appear under a microscope. As you can see, this is a lymphocyte. The nucleus of a lymphocyte is so big and round that it is almost covering the whole of the cell. The color of this nucleus is blue, giving a pink spot appearance. This is a monocyte. The nucleus of a monocyte is dark blue and kidney shaped. It is present towards the periphery of the cell. This is an eosinophil. The nucleus of an eosinophil is spectacle shaped. It is bright red colored and bilobed. The connection between these lobes is slightly thicker. This is a basophil. The nucleus of a basophil is blue colored and bilobed. The connection between these lobes is very thick, which is not clearly seen, as you can see here. This is a neutrophil. As I have mentioned earlier that neutrophils are polymorphonuclear, it means multiple lobes are present in their nucleus. The connection between these lobes is very thin. The color affinity of neutrophils is neutral, that is a combination of both red and blue colors. When you place the slide under a microscope, cells will appear like this. This is your first field. Now draw 100 squares on a paper and fill these squares with whatever type of white blood cell you see in this field. As you can see, all types of white blood cells are present in this field. So, you will write B for basophil in first square, M for monocyte in second square, L for lymphocyte in third square, N for neutrophil in fourth square, and E for eosinophil in fifth square. Now you have mentioned all types of white blood cells that you can see in this field. Now what you are going to do, you are going to change your field. For suppose this is your second field where you can see only two neutrophils. So what you are going to do, you are going to write N for first neutrophil in this square and then again N for second neutrophil in another square. Now again you are going to change your field. For suppose this is your third field where you can see only one neutrophil. So you will write N for this neutrophil in another square. You keep on changing fields until all these 100 squares are filled. Then you are going to add the amount of each type of white blood cell separately to get the total differential count of leukocytes. Let's suppose after adding the amount of each type of leukocyte separately, you get this result. Adding them all will give you a total count of 100. What is the clinical significance of this practical? The percentage of neutrophils increases in acute bacterial infections and inflammation. The percentage of eosinophils increases in hypersensitivity reactions and allergy and also in warm infections. An abnormally high level of basophils is called basophilia. It can be a sign of chronic inflammation in your body. It can be caused by infections, severe allergies, or an overactive thyroid gland. Normal count of lymphocytes is 25 to 30%. Their number increases in chronic infections such as tuberculosis and viral infections. These lymphocytes produce antibodies and cell-mediated immunity. High levels of monocytes may indicate the presence of chronic infection, an autoimmune or blood disorder, cancer, or other medical conditions. What are the precautions? Pricking should be done aseptically. The slides should be cleaned, dry, and free from grease, and movement of the second slide over The first slide should be smooth. Here is your study question. What is the special importance of blood smear?